Hello champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 day series, and today we are going to talk about the very next chapter, which is molecular basis of inheritance. Again, a high priority chapter along with principles of inheritance and variation. And I thought let's start with basics because it is very much important. So we will be talking about the structure of DNA and also the discoverers who were behind the structure, who helped to find out this particular structure. Now what did we cover in the previous session? We spoke about principles of inheritance and variation. The chapter is completed. So in total, within 30 days, 34 days, we have completed 5 chapters. Okay, one is reproduction, human reproduction, reproductive health, and then sexual reproduction in flowering plants, and then we spoke about human health and diseases, and this was the 5th one. And now we are going to start off with the 6th one, which is a very good thing, right? Now I said 6 questions were asked the previous year, but it was more than 6 the previous years. So we can never imagine what could happen the next year as well, which is why I'm saying you have to be very well prepared, and which is why I thought I'll divide this chapter into small chunks in ways that it will be easy for you to remember at the end. Okay, so this is the thing. Now going ahead, let's understand the basics over here before getting into the things. So you need to know something that is known as central dogma. And what is central dogma? Basically, central dogma says that DNA is getting converted to something, and that something is getting converted to something. It's not getting converted. Basically, the DNA helps to make the proteins with the help of this something. Okay, you don't understand? It's okay. Over here, we have a DNA. And we studied in a previous class, this DNA has certain segments, and these segments, what do you call them? You call them genes. Okay, and these genes, what are they responsible for? Ma'am, they are responsible for making proteins. Okay, so basically if imagine there is gene 1 over here, and this gene 1 will be responsible for the formation of protein 1. Going ahead, think of there is gene 2, and this one will be responsible for the formation of protein 2. Similarly, you have so many different proteins being made with the help of genes, and that is why we are the way we are looking right now. Okay, all our physical characteristics are determined by the proteins that are there in our body. But is this a direct process? Can DNA directly help in the formation of proteins? No, children, it's not a direct process. Okay, the DNA has to change itself into ribonucleic acid. So does it totally change itself into ribonucleic acid? No, it's not like that. Basically what happens, suppose this is a DNA, I just uncoiled it. So now it is looking like this. I just uncoiled it, now it is looking like this. This DNA, whatever information is there in this DNA, it is transcribed to form an RNA, and this RNA you call as mRNA. M stands for messenger RNA. Okay, M stands for messenger RNA. So whatever information was there, it is being transcribed, and this process is known as transcription. This process is known as transcription, where whatever information is stored here is being put down to a new strand, that is mRNA strand. Okay, mRNA stands for messenger RNA. So did anything happen to the DNA? No, DNA just gave the information that you have to do this, that's all. Okay, and that information is stored in the form of mRNA. But why is this happening? Why can't DNA directly make proteins? Because there is a catch, and the catch is that in our cell, you know, there is something that is known as nucleus, and inside our nucleus resides our DNA. Why can't it be outside? Because outside there are so many reactions happening, there will be so many enzymes, and these enzymes or these reactions could have an impact on the DNA. And you have studied in the previous lesson that even one single mutation in our DNA can lead to some of the other diseases. For example, sickle cell anemia, it's caused like that only. So can we take the risk of sending our DNA outside to this region, which is too much risky? No, we cannot. And why are we actually sending things outside? What is in there outside? So to make proteins, there are certain special organelles, and you call them ribosomes. What do you call them, children? You call them ribosomes. So the DNA has to meet ribosomes for the formation of proteins, or else it cannot. So DNA thinks, since I cannot go outside, it will be really risky, I'll actually transcribe. Please do not write conversion. There's no conversion happening. DNA is as in like that only. It's just providing information for the formation of an mRNA. That's all. Okay, and this process is known as transcription, where the DNA is helping in the formation of RNA. Okay, now transcription is being done such that this messenger can take the message and this mRNA is fine to go outside. So it goes outside and this message is read by the other organelle, that is ribosome, for the formation of what, children? Protein. Okay, so when this message that is there in the mRNA is getting translated to proteins for the formation of proteins, you call this process as translation, where you are translating the message which is there that you have to form this protein, guys. Okay, so DNA to RNA, what do you call this process as? Transcription. RNA to proteins you call as translation. Can this happen? We cannot send our DNA outside. Who is there outside? Ribosomes. Ribosomes are the one which reads the RNA and makes the proteins. Okay, so this is what the central dogma says, that DNA basically is transcribed to RNA and the RNA is translated to proteins. Well, this central dogma was given by none other than Francis Crick. What was his name, children? His name was Francis Crick. Done? Easy? So will you remember the central dogma? I hope that you remember this particular very important thing. Okay, this has been asked so many times. Going ahead, we need to learn the structure of DNA. So in this chapter, transcription will be in detail, translation will be in detail, everything will be taught in detail. Okay, so as of now we are just tune on into the basics, right? So DNA structure we need to study. So let's study that. So DNA is basically a chain of nucleotides. What is it? A chain of nucleotides. So basically you can just imagine beads over here, or basically you can just imagine that there is one nucleotide and this nucleotide is attached to another, which is why I call this as a polymer of what, children? Nucleotide. Now in DNA structure you will be hearing so many things: nucleoside, nucleotide. The first thing that comes is nucleotide. Always remember, tides are very big. Similarly, our DNA will be very big with the help of these chain of nucleotides, or you call polymer of nucleotide. Now what is nucleotide made up of? Okay, one particular unit like this is made up of something, and the first thing that it is made up of is known as pentose sugar. So pentose sugar basically I have made over here, but for your understanding I'll show. So over here you have a structure like this. Okay, you name this as 1, this is second position, this is third, this is fourth, and there is one carbon attached over here, so this is fifth. How many carbons we have? 5, which is why I call this as pentose sugar. Pent as in 5. Okay, this is a pentose sugar. Fine, agreed. Now on the second carbon, if there is OH group attached on the second carbon, you call this whole sugar as what, children? You call this whole sugar as ribose sugar. Okay, and if you are asked the difference between DNA and RNA, this is one difference, because RNA has this type of sugar where on the second carbon OH group is attached, and so their basic pentose sugar is what? Ribose. On the other hand, when you talk about DNA, things are quite different. As you see over here, I have cut down the O, so there is no OH group. Instead of that, there is just one H. There is just one H, children. Okay, and you call this whole sugar as deoxyribose sugar. What do you call it? Deoxyribose sugar. So you know the first thing that the nucleotide is made up of is a pentose sugar. It has 5 carbon. 1, start with this side. Okay, always start with this side, that is your left side. So 1, 2, 3, 4. Okay, so imagine the hand that you do not use very much. 1, 2, 3, 4, and then one thing is also there, 5. Okay, the one that is there, it's 5. Now if it is a ribose sugar, it will have OH group on the second carbon. If it is deoxyribose, it will not be having O, which is why it is known as deoxyribonucleic acid, whereas RNA is known as ribonucleic acid. Both of them, both DNA and RNA are nucleic acid, children. Okay, they might have a acidic group as well, which is why you call them acid. Okay, done. Let's go ahead. Next structure which it is made up of. Okay, so this is a pentose sugar, and over here your in your DNA your H was there. So I'm not writing because everyone has H over there. So it's not something uncommon. So we are not writing over here. Okay, H is just there. Just imagine if OH group is there, we just definitely put it OH. Okay, but here we are talking about deoxyribose sugar, which is there in the DNA. So we don't need to put. Now, apart from the pentose sugar, you see pentose sugar is attached to something else, which is what, children? Nitrogen base. Oh, which is what? It is known as the nitrogen base. Fine. And where is it attached? On the first. So you can remember like this. So there is something like this. We look like this. Over here also it is attached to something else, and over here also probably it's going to attach with something else. But let's stop here itself, okay, and think. So we have pentose sugar, which is the whole structure, and on the first carbon we have a nitrogen base. Okay, we have something that is made up of nitrogen. You can think like that. So we call it what? Nitrogen base, children. Now I say A and B structure, the whole thing that you see as of now, I have a name for it, and that name is nucleoside. I told you nucleoside, nucleotide, everything will be there. So we have not made nucleotide until now. We're still in the making process itself. As of now, we have got something that is known as nucleoside, which has the nitrogen base and which has the pentose sugar. Done? Now, yeah. So first thing, since we are in this way, let's talk about nitrogen base also first. There might be different types of nitrogen base, right? So we need to talk about that first, and then we'll go ahead with nucleotide. So nitrogen base, if we talk about DNA's nitrogen base, we have two types of nitrogen bases. One you call them purines, other one you call them pyrimidines. Okay? One you call them purines, other one you call them pyrimidines. Now over here, under purine comes adenine, and the other one that comes is guanine. Adenine and guanine. Now, adenine says, I am best friends with thymine. So, under pyrimidine comes thymine, and thymine and adenine are the ones that are always together. Whereas, guanine says, even I have a best friend, okay? And his name is what? Cytosine. So, there is cytosine, which is always linked to guanine. So, doubt, ma'am, can't this guanine and adenine come together? No, children, they kind of hate each other. They do not want to come together at all. So adenine always pairs up with something that is known as thymine, whereas guanine always pairs up with something that is known as cytosine. But I have to tell you one more thing. That is, the bond between guanine and cytosine is kind of more stronger because they share 3 hydrogen bonds. These bonds that you see are nothing but hydrogen bonds. Okay? So there are 3 hydrogen bonds in the case of guanine and cytosine, whereas as you can see over here, adenine and thymine has 2 hydrogen bonds. Now, ma'am, something is written over here. Okay, I'll talk about it. So, nucleotide bases or nitrogen bases, how many of them are there? Okay, you can just remember, if you're not able to understand nucleotide, we have not come until nucleotide. So, just remember nitrogen bases. How many of them you have? Purines and pyrimidines. So, adenine always goes with thymine. Guanine always goes with cytosine. Okay, will you be able to remember it? A is the first letter and T comes to be very end. Okay, so you can remember like that, A-T. Fine. And it has just 2 hydrogen bonds. Over here, it has what, children? It has 3 hydrogen bonds. Now, again, there is some difference between DNA and RNA here also. In pentose sugar, we saw the difference. In here also, they have some difference. That is, in the case of RNA, okay, they also have purines and they also have pyrimidines. Fine. Example of purine is adenine, and the other example is what, children? Guanine. A-G. Okay. Over here, they have guanine has cytosine. Okay, but adenine is kind of sad because her friend is now changed from thymine to something else, that is uracil. So now adenine will be pairing up with uracil, guanine will be still the same, it will be pairing up with cytosine. So in the case of RNA, they do not have thymine, children, they have something that is known as uracil. And RNA was the first genetic material that we discovered. Okay, initially RNA was something that was responsible for formation of DNA to some extent when you study evolution. So the basic thing that initially came was uracil, but since uracil was not that stable, it was replaced by something else that is known as thymine. All this thing that you are seeing over here, you call them what, children? Nitrogen bases. What do you call them? Nitrogen bases. So in total you have 2 pyrimidines and purines, okay, purines and pyrimidines, and under purines comes adenine and guanine, under pyrimidine comes thymine and cytosine. Okay, over here also you have uracil and cytosine. So in total, if you are asked about how many pyrimidines are there, okay, so this is common, right? So cytosine is 1, okay, thymine is the next one, and uracil is the next one. So we have 3 pyrimidines, whereas when you talk about purines, in total we have 1, and this is what, children? 2. We have 2 purines. So purines, A is the, okay, purines, the word is shorter, okay, whereas pyrimidine, the word is longer, so it comes later. You can think like that. Okay, it comes later, and A is the initial word, so you can think like A comes before. Either way you want to remember, whatever way you want to remember, you can remember. Okay, will it work for you? Now, I wanted to tell something very much important. Yes, okay. So, if you talk about DNA, this DNA basically, you saw that there are something like this. What is this something? What is this something? Basically, the whole thing, the sugar that you are going to see in the nucleoside, everything is going to be arranged now, and there will be nucleotide, so nitrogen base over here and nitrogen base over here. Similarly, one nitrogen base over here, another nitrogen base over here, another, and these, they bond. If it is guanine and cytosine, there will be 3 bonds. If it is adenine and thymine, they will be having 2 bonds. So in the case of DNA, this is a very important piece of information. In the case of DNA, okay, the amount of pyrimidines is equals to amount of purines. Didn't get it? So just imagine, if A is there, T is there, right? A is purine, T is what? Pyrimidine. If G is there, C will be there. G is what? Purine. C is what? Pyrimidine. So for every purine there will be pyrimidine, right? So pyrimidine and purine number will be same. Agree? So I say 50%, write it somewhere, 50% of DNA is equals to purine, and children, next 50%, okay, of DNA is pyrimidine. Will you write it somewhere? 50% of DNA is purine, 50% of DNA will be what? Pyrimidine. Fine. Now, one very important thing. Should I teach it now or later? Okay. As of now, you can remember this. Okay. I will give you a small question. I'll give you a small question which will make you understand things better. So, I am saying guanine, okay, guanine is 25%. Is 25% in the DNA. What is the amount of cytosine? What is the amount of cytosine? That is the first question. Next, what is the amount of adenine? So, if guanine is 25%, obviously it will have cytosine also on the other hand. So, that will also be 25%, right? And now, if I am asking about adenine, if I am asking about adenine, if guanine is 25% and I said 50% of the DNA is purine, okay, out of 50%, if 25% of purine is guanine, there will be another 25% which will be what, children? Adenine. So, this will also be what? 25. Now, if I change the number, okay, please do not get confused. If I change the numbers to something else, probably 15. If I say 15% of the DNA is guanine, then what about cytosine? Similarly, it will also be 15%. But what about adenine? This time you have to minus 50 minus 15. What do you have, children? You have 35. 35% of this will be adenine, obviously. And if you are asked about guanine, obviously, adenine, and you asked about thymine, then adenine pairs up with thymine, so that will also be 35%. Okay, cool. This is how you have to solve the questions. It's a very important thing. Okay, so we know nitrogen bases are of two types, purines and pyrimidines. Adenine and guanine is over here. Pyrimidine, if you talk about that, thymine and cytosine is over there. Okay, A and T has 2 bonds, G and C has 3 bonds. Done. Over here we have a pentose sugar, and this pentose sugar has a base which is known as nitrogen base. Okay, it is attached to nitrogen base on the first carbon. This whole structure you call it as what, children? You call this as nucleoside. Now let's see how to make it a nucleotide. It's still not a nucleotide. So let's see how can we make that. Over here, if we add a phosphate group on the 5th carbon, I told it will be like this, right? So on the 5th carbon, if I am putting a phosphate group, this whole structure becomes a nucleotide. So phosphate plus nucleoside becomes what? A nucleotide. This and this, if we remove this and we talk about other things, then it was a nucleoside, right? If we add phosphate group, it becomes what? A nucleotide, children. So please remember, start from here, left side, 1, 2, 3, 4, 5, and on the 5th carbon you have phosphate group, on the 1st carbon you have nitrogen group. Done? Now, you have to remember the name of the bonds as well. It's not like chumma is there. So the first carbon is bonded to nitrogen base, and this bond that is being made, it is known as glycosidic bond. What do you call it? Glycosidic bond. On the other hand, when you talk about the 5th carbon and the phosphate group, this is known as phosphoester bond. What do you call it? Phosphoester bond. Please do not get confused with phosphodiester and phosphoester. This is just phosphoester. When there are there are two ester bonds, you call it phosphodiester. What do you mean by ester bonds? If there is any chemistry fans over here, you can tell me what do you mean by ester bonds. You might be already studying about it, right? When there is a bond between OH and what? I said, tell me. Anyways, so we have 2 bonds. One is glycosidic bond over here, the other one is phosphoester bond. Now let's make one over here. How can we number from the left side? So this 1, 2, 3, 4, and then one there is 5. Agree? On the first one, what is attached? On the first one, you have a nitrogen base attached, and this bond you call it what, children? You call it glycosidic bond. Okay. Then on the second carbon, you're leaving it empty. In deoxyribose, there is just H. So just drop it like that. 4th, okay, chodo. 5th, we have another one which is known as phosphate group is there, and this bond is known as phosphoester bond. Okay, we do not have any one over here as of now. Now things are going to change. This whole structure is known as nucleotide, and now things are going to change a bit. Let's see. So over here we have to arrange them, because I told you that nucleotide, they are the monomers. Nucleotide are basically the monomers which are going to come together. When monomers come together, what do they make, children? They make a polymer. So if you are asked what is the monomer of DNA, it is nucleotide. Don't write nucleoside, I'll kill you. So it is nucleotide, it is the monomer. Okay, so many monomers come together to form what, children? The polymer. Cool? Works. Now over here I'm going to arrange all of them, but can they just be in the air? There has to be something that links them, right? There has to be some very important factors that links them. So let's see what is that. So this phosphate says, hey, I kind of like you so much. It's talking to the other one. Okay, kind of like you so much, I want to get attached to you. Okay, and the other one says, fine, accept it, it's fine, it's all good for me. So this phosphate goes and attaches to the nearest carbon, that is the 3rd carbon. What is happening? The phosphate which was attached to the 5th carbon, okay, it is now saying that I want to attach to the next sugar, so I will be attached to the 3rd carbon. Okay, now over here also same thing happens. This phosphate is attached to the 3rd carbon, and in this way, don't you think a chain is being formed? Yes, a chain is being formed, right? Now over here, this particular bond, what were you calling it? You were calling it phosphoester bond, right? Which was there in between this. But now you have 2 such things. Here also there will be carbon, here also there is carbon only. and it's all different. It's in the bond between this and those things that are present over there. It's not exactly carbon. Basically, there is another thing over there present. I'm not saying that. As of now, just understand that both of them comes together. So this is phosphoester bond, this is also phosphoester bond. So now it's time for renaming them. It's time for renaming them. I'll call the whole thing as phosphodiester bond. Di as in 2. So this is what, children? Phosphodiester bond. And if you see, I have made all the things just like that. First is having nitrogen base, and then fifth is having phosphate. Sorry. And this phosphate is getting attached to that one also. So your ma'am is so frustrated that she is hitting herself. Yes. So this is how the chain is being formed. But you know, if there is adenine over here, then it needs a partner, right? And the partner is what? Thymine. So thymine should be there on the other side. So now the structure should be looking, okay, I've made the same thing over here. Now the structure will be looking something like this. But if you have noticed, there is some difference. Okay, if I made things like this, just a second. Children, are you enjoying the class or is it too boring for you to learn? Do tell me. Okay, so over here, if this was the first, this is second, this is third, this is fourth, this is fifth, right? On the first, NB was there. And if I make just something like this over here also, the NB will be on the other side. It will be like, take that, right? That situation will be happening over here. So we do not want that. They are friends. So this has to turn this side. So I cannot do that. Obviously, it will be always attached on the first carbon itself. So what can I do for getting them attached? If I just turn them around, then I think the structure will be something like this. Okay. This will be the first one, second, third and fourth. Now here I will be having NB, similarly here also I will be having NB, and both of them can attach. So I just turn it up. So one will be like this, other one will be like this. You call it as anti-parallel. Are they parallel? No. One is going this side, the other one is ulta. You call it what, children? This structure of DNA, please remember. Structure of DNA is anti-parallel. Fine. Now, over here, the nitrogen bases which are there will be having hydrogen bonds. You know that 2 and 3 depending on what they have. Now, what is a new addition that we have? So you can think same thing is happening over here. Fifth carbon, you have phosphate, and the phosphate is attached to the third carbon. So it's going on like that. Now, but obviously it will have some end. It will definitely have some end region. Let's see what happens in the end. So over here, there will be one phosphate which will not have anyone to go and pair with. It's a sad thing, right? And it's on the fifth carbon that this phosphate is attached. So I call this particular end as 5' end. What do I call it, children? 5' end. Is that important, ma'am? Very much, children, because we are going to study about transcription, translation, everything. So we need to know about direction. There it cannot go from left to right and things like that. We say from 5' to the next dash end. What is the next dash end? Basically the 3' end. So you might be seeing something which you were not seeing until now. That is, there is an OH group on the third carbon, and that is visible over here. So basically, usually what was I was telling you, phosphodiester bond is made. Right, phosphodiester bond is made. Basically, when ester bond is made, it's between OH group and an acid. So basically, this C might be attached to an OH group, and this OH group is basically attached to phosphate. Similarly, over here also, you have some OH group, which is why I call it also ester bond. So there are two, so I call it phosphodiester, diester. Now, similarly, this time, there was no one to pair over here as well. So I put OH like that. Simple. So this end where OH is there, okay, I call this as 3' end. What do I call it? 3' end. Now since we twisted everything this side, so here also this phosphate is like, I do not have everyone. So it will be 5' end, and they are anti-parallel. So if there is one 5' end there, the other 5' end will be here. It cannot be there, right? So this is the 5' end. On the other hand, there you will be having the 3' end. So you have 5' end, you have 3' end. 5' why I am telling you again, because the 5th carbon has the phosphate, which is why you call it 5' end. Over here 5' end. Over here you have 3rd carbon which has OH group, and it will be known as 3' end. There is no one to pair. Both of them are really sad. So one strand goes from 5' to 3' end, then the other strand goes from 3' to 5' end. Will you be able to remember it? They are anti-parallel, children. They are anti-parallel. Okay? These bonds are hydrogen bonds. And with this, we understood the basics of DNA, the basic thing that you need to know. Now, one more thing, okay, when you talk about a DNA as whole, okay, when you say this to this, it's not like there is one turn and there is second turn. No, this whole thing from here to here, whatever you see, you call it one turn or one loop of DNA. So whole thing. So it is made up of 2 smaller segments. You can think like that. Okay. So this whole thing is one turn or one loop of DNA. Just think that it's going something like this. Okay. So whole thing is one loop of DNA, and each loop of DNA will be having 10 base pairs. You want to know? 1, 2, 3, 4, 5. Hey, ma'am, what is base pair? Basically, when the nitrogen bases come together, you call this a pair, and since it is made up of nitrogen bases, you call it base pair. You call it what? Base pair. So, how many base pair do you have? 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. You have 10 base pair in one turn of DNA. Fine. And you also call the whole distance. If you want to know the distance, you call it the pitch of DNA. What do you call it? Pitch of DNA. This is given in your NCERT. It's not something that I am making up. Okay, it's given in your NCERT, and you call it pitch of DNA, the whole length of DNA. And what is the pitch of DNA? It's 3.4 nanometers, or nanometer if you convert to Armstrong, it is 34 Armstrong. Will you be able to remember it? What is the pitch, children? 3.4 nm. That is the pitch, that is the whole distance, okay? 3.4 nm is 34 Armstrong. Now, if you're talking about the distance between each base pair over here, I'm not talking about their distance, I'm talking about this and, okay, this and another base pair's difference, this much. What will it be? So, if you have 10 of them and the whole length is 3.4, then 3.4 divided by 10, it will be 0.34 nm, children, nanometer. And if you talk about Armstrong, then it will be 3.4 Armstrong. Okay, so please not get confused if they are asking about the pitch and they have given the option as 3.4 Armstrong, 3.4 nm or 34 something. Please remember it's 3.4 nm. Okay, not Armstrong. Please do not go and write Armstrong and come there. Okay, 3.4 nm. 3.4 nm is equals to 34 Armstrong, whereas 0.34 nm is equals to 3.4 Armstrong. This is what, children, pitch of the DNA. Okay, this is pitch of the DNA, but that is given over here. This is the distance between each base pairs. Okay, fine. Space between each pair, that is this. Now let me hide and ask you once more. What is the pitch of the DNA? This is the length. What is the length? 3.4 nm. Now this 0.4 nm, if you want to convert into Armstrong, it will be 34 Armstrong. Okay, this is the pitch. If you are asking for the distance between each pair, then it will be what? It will be obviously less, because you have 10 of them in this. Okay, you have 10 base pairs, so 3.4 divided by 10, it will be 0.34 nm or 3.4 Armstrong. Works? Will you remember it? It's again a very important piece of information. So I hope you understood this as well. Cool? Now, we need to know the names of the people who were behind all this thing, the discoverers, who were behind the discovery of DNA. So there was a man whose name was Frederick Mischer. What was his name? Frederick Mischer. Okay, Mischer that you eat. You remember like that. So Frederick Mischer, sometimes you would find its taste is something like different, some acidic, some sour taste, I don't know what. Even though it does not taste like that, you have to remember it like that. So Frederick Mischer saw an acidic substance in the nucleus. Okay, And he thought that probably this structure has something to do with our genetics. Okay, and he called it nuclein. What did he call it? Nuclein, because he found it inside the nucleus. So Friedrich Mischer was a person who said that there is some acidic substance inside the nucleus, and I am going to name it what? Nuclein. And this was done in the year 1869. Mendel did his studies in from 1856 to 1863. We studied in the previous chapter, right? So 3 years after that, Friedrich Mischer, who was there, he found that okay, there is some acidic substance in the nucleus, and I can call it nuclein because it's there in the nucleus. Okay. Now, while all this was happening, there were other people who were working. Okay, there were other people who were using all the experimental stuffs and things like that. So the other people includes Maurice Wilkins and Rosalind Franklin. Okay, Maurice Wilkins and Rosalind Franklin used X-ray diffraction data. Okay, they did X-ray diffraction data to find out the structure of DNA. Okay, Maurice Wilkins and who? Rosalind Franklin. And then came James Watson and Francis Crick in the year 1953. You see the difference, almost like more than 100 years, right? More than 100 years is happening here itself. So basically, not 100 years, not more than 100 years, but near to 100 years, yeah. So when Mischer said that, okay, there is some acidic substance in the nucleus, everyone like, no, who cares, okay, and it was nuclein. But then in 1953, okay, in 1953, things kind of changed. What happened? Francis Crick and James Watson said that I have a structure to show, we have a structure to show, and that is DNA double helix structure, as in this is one strand, this is another strand, so double. Okay, helix as in when there is a turns and things stuff like, you call it helix. So DNA double helix model was given by James Watson and Francis Crick, but they would have been it would have been a failure if they did not get the data from this particular research that was done by Maurice Wilkins and Rosalind Franklin. Okay, if you remember, Francis Crick was the one who was responsible for putting down the central dogma that DNA forms the RNA, RNA forms the protein. Okay, now this thing we will study, but as of now I want you to remember them. That is much more important for me as of now. So let's start studying. Okay, we have Friedrich Mischer. Okay, he saw an acidic substance when children 1869, he saw acidic substance which he named as nuclein. Okay, while this was happening, there were other people whose name was what, children? Rosalind Franklin and Maurice Wilkins. Okay, you can remember RM, regional manager. They were working on this region of DNA and stuff like that, and with the help of X-ray diffraction, they put down a data. With the help of this technique, that is X-ray diffraction, they put down a data. Okay. So the RM, regional manager, worked, Rosalind Franklin and Maurice Wilkins worked, and they put down this particular data. But then someone was there who was working on this thing. They used this data. Who were they? James Watson and Francis Crick. They said, if we use this information, probably we would be able to deduce something that is known as DNA double helix structure. And when was this done? In the year 1953. So in the year 1953, this was done by them. I hope you remember. Please remember RM. Always the problem that people used to do is they confuse with Franklin. Franklin they think of as a first name and get confused with Francis. Franklin is different, Francis is different. So RM, Rosalind, you can remember, right? And Maurice, RM. Okay, forget Franklin for time being, if you want. Okay, and they did the X-ray diffraction, with the help of which they put the data. This data was used up by James Watson and Francis Crick, who found the DNA double helix structure. okay, who put down the DNA double helix structure. But who was the first one to see that there is something like this? Probably it was Frederick Mischer, but he was not able, okay, in his mind everything was mixed. He was not able to understand what to do. I don't know what is this, okay, because it was lots of years back. Okay, so you can remember in Frederick Mischer's brain everything was mixed, and that's what he told that I do not know about DNA and all. DNA came very late. Okay, let me just name it as nuclein. So he named it as nuclein. Cool. Now, while all this was happening, there was another scientist whose name was Erwin Chargaff. What was his name? Erwin Chargaff. Okay, and Chargaff's rules, laws and everything is there. According to Chargaff's laws or rules, basically the ratio of adenine and thymine, okay, guanine and cytosine will be same. That means obviously if you have adenine over here, as I said earlier, thymine will also be there. So the ratio of this and this will be same. Similarly, if G is there, C will also be there. So ratio of this and this will be same. Okay, agree? So can I say, okay, if I want to write it somewhere, well, where will I be able to write it? Okay, here I'll write it. Please don't see the question as of now. Let me just explain you something. Okay, so over here, Chargaff said ratio of purines and a U and pyrimidines are same. That means A is equals to 2 and G will be is equals to C. This is purine, okay, and this is what? Pyrimidine. So this is purine, I am writing PU, and this is pyrimidine. Fine. Now the second thing that he said that A cannot be equal to G, and obviously T also would not be equal to C for obvious reasons. Can I say that it would be always equal? No. I just said that purines and pyrimidines can be same. So basically if I have 50% of purines, I will be having 50% of pyrimidines. Now in this 50%, I am saying 20% is basically, okay, 20% or 20% if I am saying it is adenine. So obviously 20% it will be guanine, thymine as well. But this is thymine, this is adenine, okay. But do you think that guanine can also be 20%? No children, guanine cannot be 20%. It will be 30% because 30 plus 20 is equals to 50, right? 50% should have been purine, 50% should be what? Pyrimidines. So it will be 30. Now C can be 30. So A and G, are they equal? No children. A and G, they are not equal, not T and C can be equal. Please understand. So ratio of purines and pyrimidines are the same that they have written. Second thing, they are also saying that this purine and pyrimidine number, okay, if you see two purines together, their number would not be same. Their number cannot be same. That is what written over here also and here also. Okay, am I clear? So A plus G plus T plus C cannot be equal to 1, right? Both purines when they come together and pyrimidines when they come together, it cannot be equal to 1. Please understand that. Okay? Clear? Ayo. Did I mess it up? No. Guys. Sorry. Purines, if you take to pyrimidines, it will be equal to 1. Okay, purines is and pyrimidine, it will be equal to 1 because the ratio is same. Please remember like that. Sorry. Didn't want to make you confused. I'm literally sorry. So this is what Chargaff said. Erwin Chargaff was the one who put down this statement. Okay, so while finding the DNA health structure, they also got to know the reasons. Probably because there are nitrogen pairs and they are attached to each other, which is why there's a DNA double helix that is being formed. Please remember the name of all these people because it is very much important. Now, we'll solve the first question. Please try to solve it over here itself. The diagram shows an important concept in genetic implication of DNA. Fill in the blanks. So, A is what? A is this, where DNA is forming the mRNA. B is this, where mRNA is forming the protein, and this thing, whole thing was proposed by whom? That is C. Tell me. So A is what, children? When DNA is giving its information to mRNA, you are not translating the information. Translation is done baad mein, later on, right? So initial thing that comes is transcription. So A has to be transcription, B will be translation. It was proposed by none other than Francis Crick. So this is the correct answer. This came in NEET 2013. Easy question, no? Yes, something very similar to this I saw. Okay, in a sea urchin DNA which is double stranded. Okay, in sea urchin DNA which is double stranded, 17% of the bases are shown to be cytosine. Okay, 17% is, children, C. The percentage of other 3 bases expected to be present in this DNA are. So if C is 17, who is pair of C? G. G will also be 17. Okay, if G is 17, okay, I am writing like this: A, G, T and C. So C is 17, so G is also 17, fine. But if this is 17 and the whole thing it has to be 50, then 17 minus 50, sorry, 50 minus, yeah, whatever you do, you get what, children? 33. So 33% will be adenine. Now if A is 33, so this should also be 33, because all the A will be pairing with T, right? Now let's see. So G is 17, A is 33 and T is 33. G is 17, A is 33, T is 33. This is how questions come. So please be assured that you have to practice certain small things. Okay. Next thing, in DNA molecule, the proportion of adenine in relation to thymine varies with the organism. There are 2 strands which run anti-parallel, one in 5' to 3' end direction and the other in 3' to 5'. That's true, okay, that's true. The total amount of purine nucleotides and pyrimidine nucleotides is not always equal. Is it like that? Is it like that? The total amount of purine nucleotides and the pyrimidine nucleotides is not always equal. It cannot be like that. Purines will always be equal to pyrimidines, total number. There are 2 strands which run parallel in 5:3 direction. No, one goes in that direction, other goes in this direction. The proportion of adenine in relation to thymine varies with the organism. No, adenine and thymine, that has to be same, right? So this is wrong. What is correct, children? This is correct. Did we have to find the correct thing only? I don't know why the question is like half. Okay. Anyways, this is the correct one. That all thing that you can see is wrong only. So it's fine. Now going ahead. 1 turn of helix in a B-form DNA. Okay, that is B DNA, there is A DNA, there are different forms of DNA. Okay, and the common structural DNA we call it as B DNA only, about which we studied. So they are asking you 1 turn of helix in a B-form DNA is approximately equal to what? 1 turn, that is 1 total thing. 1 loop is equals to what, children? The pitch they are asking. They are asking for the pitch. It is 3.4 nm. This came in 2006. Okay, one more. Which of the following statement is correct? Adenine pairs with thymine through 2 H bonds. Adenine pairs with thymine through 1 H bond. Adenine pairs with thymine through 3 H bonds. Adenine does not pair with thymine. Ayyo. No, children. Adenine pairs through thymine with 2 H bonds. Please remember, it's thymine and not thymine. This is different. This is vitamin. This is something different. This is the thing. Okay. And they had 2 bonds. Cool. Now these were the questions that we did. Easy, 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 right? Now everything is easy. The answer for this was this, 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 and the answer for this was this. I'll just check for this one once, okay? So that's all. And now, oh, I might have a repeated, this is a repeated question, okay? I have already completed it, okay. Now one question that will be there for you, and that is, children, this. Try this out. This is also very much easy. Okay, it's very easy. Next lecture we will be studying about how the DNA is arranged in the nucleus. DNA is pretty long. You can just imagine if one loop is just 3.4, then it has to be really, really big. So basic information is about DNA. and few more things, few more experiments we'll be studying in the next section as well. Okay, works for you? So that's all, children. I hope that you were able to understand what I taught you, and if you were able to understand, then do not forget to subscribe to the channel, and I wish you all the best for your journey. Please be motivated, please keep smiling. I just want you to be happy always, always, always. Okay, so that's all, my littlest champion. I hope that I'm not okay. For this thing, I want you to write it as homework and tell me in the comment section, okay, what exactly is it. Fine. This thing I want you to write somewhere and put it in your desk, because this thing is something that you might forget. The names are kind of confusing, so have your own tricks. If you know certain tricks, then put it down in the comment section as well. I'll be really glad to see that, but we need to remember it. With the years, if you're not able to remember it, you might lose so much of your marks, which I do not want. So that's all. Now I won't take any more of time. That's all, children. Remain blessed. Bye bye. Take care.